Good afternoon, everyone, and welcome to Under the Microscope, the Future of Food. This is part of our new webinar series from the College of Biological Sciences, and we're so glad you're here. I'm Jennifer Scott, Managing Executive Director of Development and External Relations for the college. We launched this series to give you, our alumni, friends, and supporters, a behind-the-scenes look at the incredible research happening right here at UC Davis. This year also marks the 20th anniversary of the college, and it's a great moment to reflect on how far we've come, but also where we're heading. And so over the past 2 decades, our faculty and students have been driving innovation, building community, and making a real impact. Under the Microscope happens twice a year, and each time we focus on a different theme that shows how fundamental interdisciplinary research in biology drives real-world impact, from human health and sustainable food systems to environmental resilience and renewable energy. It's part of why UC Davis continues to be recognized as one of the top public universities in the country, and why we're proud to be ranked among the top 40 globally for biological sciences by QS World University Rankings. Today, you'll hear from 3 of our leading experts, and after the final speaker, we'll open it up for Q&A. You can submit questions anytime using the Q&A function, and if we don't get to your question today, no worries, we'll follow up by email. To get us started, I'm honored to introduce someone who's been a driving force behind the college's commitment to education and research excellence, Dr. Mark Winey, Dean of the College of Biological Sciences. Dean Winey is a distinguished professor of molecular and cellular biology. His research focuses on centrosomes, tiny cellular structures that help organize cell division. When they don't function properly, it can lead to serious problems like cancer or miscarriage due to incorrect chromosome numbers. He's also the longest-serving dean in our college's history, now in his 10th year of leadership. He's a passionate champion for student success, creating an environment where students thrive through hands-on experiential learning, peer-to-peer support, and strong mentorship. Please join me in welcoming Dean Winey.
Jennifer, thank you very much for that introduction, and welcome to our guests today, many of you who have supported the college in the many activities that you just heard Jennifer speak about. I am Mark Winey. I'm the Dean of the College of Biological Sciences, and again, thank you for joining us. And to those of you who sent questions in, thank you for those as well. Hopefully, some of those questions will get addressed during the presentations or in the Q&A afterwards. Here in the College of Biological Sciences, we're made up of 5 academic departments and 3 research centers or institutes. 2 of those are large, the Genome Center and Center for Neuroscience, and then a really important virtual center, the Center for Population Biology. We have 120 faculty or so across those parts of the organization. And in addition to running a very well-recognized, distinguished research program with about $80 million a year of external support, we also have a current undergraduate population of nearly 7,000 students getting a variety of different types of degrees in the life sciences with us. We also have about 450 graduate students in the various graduate programs that are hosted here in the college. The college focuses on basic or fundamental or foundational research in the life sciences, working across the complete scale of life sciences, from the most minute molecular, biochemical, and structural aspects of proteins and other molecules in living organisms to grand timescales of evolution, which you'll actually hear about today, as well as across broad ecosystems. And as doing fundamental research, we are really focused on just understanding the living world and how it works, so that we have that basic information and that basic understanding as we then move on, or our colleagues move on, to try to address issues like disease states and understand how they work, or today looking at agriculture and sustainable food systems and how we can use our information that we've generated, knowledge we've generated over the years, to support ongoing efforts in the areas that you'll hear about today. As Jennifer mentioned, we have 3 distinguished faculty members who will present in in the order I'll introduce them in a moment, and then we'll have the Q&A afterwards. So our first presenter is Professor Siobhan Brady. Siobhan works in plant roots development and works to understand how plants and their root systems respond to environmental stress. And as you can imagine, in this age of climate change, this is an important issue in understanding plants and our agricultural needs for plants that are resilient. Siobhan is not only a member of the Department of Plant Biology, but she's also a member of the Genome Center, and she is distinguished as being a Howard Hughes Medical Institute investigator, one of only 3 here at the UC Davis campus and one of just a few hundred around the country. This is a very distinguished appointment that recognizes her excellence in the field and also offers significant research support for her research group. After Professor Brady, we will hear from Professor Sundaresan, widely known as Sundar. Sundar is a member of the National Academy, and he explores the genetics of plant reproduction. This has been very important work that has led to a method of cloning seeds that holds tremendous potential to revolutionize crop propagation and the global food security. And for this work, he was recognized last year with the Wolf Prize in Agriculture, which is arguably the top international prize in plant sciences and in agriculture. And then our final speaker today, Sundar is also in the Department of Plant Biology. Our final speaker today from the Department of Evolutionary Ecology is Professor Jeff Rossibera, who works in evolutionary genetics to deepen our understanding of maize and its wild relatives. This work has offers tremendous insights into agricultural practices and conservation efforts. And for Jeff's work, he has been recognized last year with the very prestigious National Academy of Sciences Prize in Food and Agriculture. Again, a prize that there's only one a year, and it was very great honor for Jeff to have won that. So after those brief introductions of our distinguished faculty, let's get started and hear from Siobhan Brady. Siobhan.
Thank you, Mark, for that really kind introduction. And I'll just add that I'm really excited at how the HHMI Award will be able to take our research in some new and transformative directions. Today, I'm going to speak to you about how plant cell development helps crops adapt to climate change. And I am really going to do that from the perspective of roots and plant roots. Why? Well, I love plant roots, and I think that they have tremendous beauty. If you're looking at the pattern of the organ as it's moving underground, to the beautiful radial symmetry of its cells, if you cut a root and look at its cross section, and then even down to the molecular patterns within their cells. I've been interested in roots since I was very young. I grew up in a small town in Canada where there were lots of steel factories, and some of the soils around us were heavy metal contaminated. Once I learned about how grass roots can assist in bioremediation of those soils, it not only gave me insight into their power, but also aided in transforming really my childhood landscape. So what is the challenge that faces us in these days of climate change, and how can we answer this from the perspective of roots? So roots are really one of the first lines of action of a plant to be able to face its external environment. So roots are responsible for taking up the majority of water and mineral nutrients to the above ground parts of the plant. And it's also involved in helping plants distinguish what is friend or foe in terms of bacteria, fungi, or even other plants. And so climate change caused by human factors is really causing dramatic and negative consequences to our ecosystems and agricultural productivity. And our goal is to not only understand how plants respond to these climate change stressors, but also understand how they've evolved in a variety of extreme environments to be able to adapt to these stresses, and then ultimately use these to be able to produce or breed more climate resilient plants. So what and how do we work on this? So plant cells have cell walls, which you may remember from your high school biology or even elementary school biology classes. This is a plant cell wall here, which is shown in yellow. And this plant cell wall, it not only encases the cell, but it also has the capacity to act as an armor to the outside world, to be able to protect it, but also to facilitate entry of those minerals and friendly bacteria. There are two different cell wall components that my lab works on in great detail. The first is shown here in pink, which is called lignin, which is a type of biopolymer. You would be familiar with it as the fiber in both paper and cotton. So lignin is one of these compounds within the cell wall that controls the entry of different nutrients. So for instance, it can control the entry of salt and also block it if there's too much of it. It can assist in the entry of nitrogen, and also, as you'll hear about today, can prevent the entry of other plants. The second substance that we work on is another biopolymer of a different flavor. It's fatty acid derived, and you would be more familiar with it as the waterproof substance in cork, like what you would find in a beer bottle or a wine bottle. Suberin is known to be associated with controlling drought responses and also pathogen resistance. So I'm going to give you a few little stories that are representative of what our lab research is from the perspective of roots and their cell walls. So first is a study of a parasitic plant that is a really major and urgent threat to our California tomato processing industry. So you may or may not be aware that California in the Central Valley is responsible for the production of 95% of the processing tomatoes in the United States and 30% of the global supply. And this supply is threatened by this parasitic plant called branched broomrape, which you can see here. So branched broomrape is able to invade its host plant, in this case tomato, but also that of other plant species, like, for instance, sunflower or cucumber. What this branched broomrape does is it exists as seeds in the ground that are so small. They're literally like little flakes of pepper in the ground. And when a plant is stressed, like if it's not experiencing enough water or mineral nutrients, this particular root, so this is a tomato root right here, this root produces this extra lignin in its cell walls. And then this is the branched broomrape, this parasite. And this branched broomrape, it basically forms a funnel, kind of like a straw, extending from the branched broomrape all the way into the host vascular cells, or the tomato vascular cells. And then it can suck out all of the water and nutrients. So the reason why branched broomrape is such a threat to the tomato processing industry is because there is no known form of resistance to this parasite. Work in our lab where we've identified mutants in plants that actually produce extra lignin, like that shown here in pink. If you produce extra lignin within these roots, this branched broomrape is not able to enter into the root. So the plant is basically blocking the parasite from entering by putting up this heavy armor. My lab also works on another parasitic plant species, in this case in sub-Saharan Africa, where with 8 partners, led by a collaborator in the Netherlands and funded by the Gates Foundation, we are trying to understand and really mitigate or prevent the interaction of this parasitic plant called Striga hermontica against cereal crops like sorghum or rice or maize. So again, striga has these little seeds that exist in the soil, also like flakes of pepper. And when sorghum is experiencing drought stress or mineral nutrient stress, it allows these striga seeds to germinate. And so this is really particularly urgent within sub-Saharan Africa, where sorghum, for instance, is grown primarily by subsistence farmers. And so these farmers need the sorghum to be able to feed their families. Providing chemical control or even fertilizer is beyond the financial means of these farmers. And so when the plants are stressed and the striga, although it has a pretty flower, attaches to the root of its host, it really completely destroys its agricultural productivity. And so as a result, people can't eat. And so this is an example of a sorghum root. Here, it's really beautiful symmetry, and striga, this parasitic plant. So our lab is focusing on how microbes can be used to prevent this interaction or this death of sorghum by striga. So we've found that when sorghum plants are grown with their full microbiome in gray here, compared to when their soil is sterilized and they have a greatly depleted microbiome, that the presence of the microbiome is actually able to partially suppress the interaction or the disease of sorghum by striga. We've been able to find that associated with the presence of this microbiome, that when the sorghum roots are in the presence of the microbiome, they produce even more of this molecule called suberin, shown here in green, and that's associated with suppression of the striga infection. And so this is one of many different modes by which we found that the sorghum root in the presence of microbes is able to reduce the infection by striga. We're also interested in how natural species can provide a source of variation to be able to breed plants to be more tolerant to stress. So we all know that water deficit or drought is a major and worsening problem. This is a wild species called Solanum pinalii, which grows in coastal deserts with minimal water and very little available mineral nutrients. This wild species is completely drought tolerant. It's pretty unbelievable. And we have found that this drought tolerant wild species produces a lot of suberin all the time, whether or not it's in control or in drought conditions. So these warm colors here represent a lot of suberin. We've recently shown that when you reduce the amount of suberin in this wild species by making a mutation, that those plants are suddenly very sensitive to the drought stress through a range of different physiological parameters that don't really matter, but that don't really matter to you. But in this particular case, it's really the suberin that's responsible for the drought tolerance. So what we're trying to do now is to try to find ways to make plants more drought tolerant by increasing the amount of suberin within this particular cell type in the root. So our ultimate goal is to really learn about and identify points within gene networks associated with these cell walls, and to be able to understand how to use these networks to have plants reduce their loss in agricultural productivity due to climate change factors. In the very short term, we want to develop branched broom rape resistant tomato prototypes. We want to identify microbes that will hopefully be sent to industry and undergo field trials in sub-Saharan Africa, and also to produce drought tolerant plant varieties. With that little introduction to our lab's work, I'd like to thank all of our collaborators. I've put two of the main collaborators here, and the members of my lab who have done this research, as well as our funders, to whom we're very grateful. So thank you for your time, and I'm now going to pass this on to my esteemed and wonderful colleague, Professor Sundar.
Okay, so I'm on, I think. Hello, everyone. I'm going to, my talk will be about uh, crops that clone themselves. And basically, it's how to make agriculture sustainable with more food production using hybrid crops. So the story starts about a century ago. Hang on. I'm trying to advance my slides here. And not succeeding. Okay. Okay, maybe now. Yes, that's it. It works. Okay, cool. So the hybrid seeds were discovered to yield a lot more than the inbred varieties. About a century ago, uh, scientists did experiments where they crossed two inbred lines, and these are uh, shown on the left side and the right side. These are ears of corn from two inbred varieties. When they crossed them, they got a hybrid. The hybrid made many more seeds. You can see this from the huge ear of the hybrid. So this led to a revolution in agriculture starting the 1930s, when hybrid corn became used in corn cultivation. So this graph shows you the impact of hybrid corn. That is, yields of corn were essentially flat for about half a century, and then hybrid corn came along, and since then, yields have been going up and up and up. So it's clearly made a big impact, and that's why it's called a revolution in agriculture. The next slide shows you an experiment that was done in these 1920s. So these are the inbred parental lines on the top. You can see that they are, this person standing here is taller than all of the plants. On the bottom left corner, you can see the first generation of the hybrid corn. So the plants are clearly much bigger, and they also produce these big ears that you saw in the earlier slide. Okay. Now, something interesting happens when you take these seeds and plant them in the field. You don't get back the same type of plants as the hybrids. You now get plants, some of which are short, some are tall, some are in between. This is all due to genetics, which I'll explain in a bit. But the bottom line is that although hybrids have high yields, their offspring do not. So farmers cannot just replant their seeds from their hybrid crops. What they need to do is to go back and buy hybrid seeds every year. And this can be expensive, especially for some crops other than maize, for which cross-pollination is technically difficult. Rice is one such example. Producing hybrids in rice requires a lot of effort, and that's illustrated by this slide from which shows an article in Science magazine about 2 years ago about this research where they show how hybrid rice is produced in USA, in Arkansas. These are helicopters that are flying over the rice field and blowing blowing air so that the pollen will be distributed over the field and allow cross-pollination to occur. Clearly, this is an expensive process to rent helicopters, and it makes the cost of hybrid rice quite expensive. And yet we could use hybrid rice, and in fact hybrids for all crops, because we could meet global demand in the next 50 years or so using hybrids. So here's a graph that shows global actual projected increases of rice, wheat, and cassava. But let's just focus on rice for now. So in pink is this pink solid line is the projected actual increase in production following current trends, and the dotted line is the projected demand. So you can see there's a shortfall of where we need about 25% more rice just to meet the demand. And you could meet this shortfall by just switching to hybrid rice, and you could do it sustainably using the same land, same amount of water and fertilizers, just by planting hybrids instead of the inbreds that we are using now. So what is limiting this is the cost of seeds, because hybrid rice is buying it every season is expensive, and much of the rice in particular is grown by smallholder farmers, and maybe 80 to 90% of the rice in Asian countries is smallholder farmers. So could we produce hybrid seeds inexpensively? Is there a method to do that? So this has been thought of for a long time, and about more than a quarter century ago, people have been looking for the possibility of finding how crop plants that can reproduce asexually. This is an article from the New York Times in the year 2000. And they thought that if they could do this, then there'd be a revolution in world food production. It was called the Holy Grail of agriculture. How do you make plants that reproduce asexually? Well, it's not as weird as it sounds, because actually it happens in nature. Many wild species of plants actually do reproduce asexually, and the common dandelion is one such example. It's a weed in our gardens, and it's actually reproducing asexually and clonally, so that each generation is the same as the preceding generation. So now I started in this problem not by working directly on it, but I came into it as a basic scientist. My training was actually originally physics and then biology, but trying to understand fundamental questions. And I'm an immigrant, like as a Siobhan, and the US offered all these opportunities to try out the ideas I was getting. So anyway, about 15 years ago, I was pondering this question of how do you get from a sperm and an egg to an embryo, in the case of humans, it would be a baby. In the case of plant, it would be a seed. In both cases, you have two very different cells that merge together to form a single cell called a zygote. And this zygote divides an embryo. So the question is, the zygote is the starting point, but how do you get from here to here with these two very different cells? So this is the question we try to answer. And what we discovered was that a gene called, in plants, a gene called baby boom 1 is activated by the sperm cell and induces the fertilized egg to divide and make an embryo. So here again is the question: sperm plus egg get a zygote, and then the zygote divides to make an embryo. Now this gene is not expressed in egg. It's shut off in egg cells, but it's switched on in sperm cells contained within the pollen grain, and it's switched on in the zygote. And this is when the gene called BBM1 makes an embryo. Okay, so what we then thought is, how about if we switch on BBM1 in the egg cell using a genetic trick? And we did that, and we found that if you did that, you could make an embryo without fertilization. So this is called parthenogenesis. So no sperm was needed, no pollen was needed to make the embryo. You just used this genetic trick. Now, to give you more in-depth thing, I'm going to play this video because that explains it much better than I could. It's produced by the Innovative Genomics Institute at UC Berkeley about this research.
Traditional hybrid breeding is a process that generates crops with high yield and other desirable characteristics. By breeding two different high-performing plants together, we can generate seeds that grow into a so-called hybrid plant that grows even better. For some crops, the hybrids can produce double the yield. Unfortunately, when the hybrid plant self-fertilizes or is bred with an identical hybrid plant, its seeds will produce plants with lower yield. This is why farmers have to buy new hybrid seeds every year. So why aren't offspring identical to their parents? Let's zoom into their cells to find out. Sexual reproduction begins with a mother egg cell. In a hybrid plant, the mother egg cell contains special versions of different genes that, in combination, give the hybrid its high yield. A process called meiosis divides the DNA into separate egg cells. The male cell, or sperm, then fertilizes one of the egg cells, and the fertilized egg begins to grow into an embryo, eventually forming a whole seed. The resulting seeds inherit different combinations of genes and will have a very different genetic makeup from the parents, just like you'd expect for human children. If the offspring ends up losing its powerful combination of high-yield genes, it will have lower yields than its hybrid parent. In this breakthrough study, Dr. Sundaresan and his team have managed to engineer rice plants that create seeds that are exact clones of the parent. The engineered plants reproduce asexually, yielding clonal seeds with all of the parent's desirable traits. So how did they achieve this remarkable feat? To enable asexual reproduction, Sundaresan and his team first used CRISPR genome editing to turn off 3 genes and stop meiosis. This produces egg cells that carry all of the mother's genes without any of the shuffling and gene loss that normally happens. Next, the researchers added a gene called babyboom1, or BBM1, which triggers the egg cell to develop into an embryo without the need for sperm, eventually producing a full seed. In the end, each seed carries a perfect clone of the original plant. This breakthrough means that hybrid plants with desirable traits will produce seeds that can be planted and grown into plants that are exact clones, with high yields year after year. Even great-grand plants will still be clones of the original rice plant. Currently, not all of the seeds from the same engineered plant are identical clones. But now that the big technical barrier has been overcome, the team is working to increase the efficiency. Excitingly, since all of the key genes involved in this approach are found in other plants, the same strategy could potentially be used in other important crops like corn, tomatoes, and wheat. Right now, most rice farmers do not grow hybrid rice crops due to the high cost of the seeds. This IGI project could transform the world, one day giving farmers in developing countries access to all the benefits of hybrid rice without having to buy expensive seeds every year.
Okay, so that video is from 2019. We've now gotten it to nearly complete efficiency, and I'll show you in a commercial hybrid rice. This is the hybrid on the left, and these are the sexual progeny shown here. They're segregating all these different types of heights and grain yields. But when we made it, when we modified the genes using CRISPR editing and the techniques that I mentioned, we now get progeny that are identical to the parent. And the impacts of this are that we can lower cost of seed production because all you do is to plant hybrids and then you get back hybrid seeds. It's very cheap to make that. And smallholder farmers can save their own hybrid seeds for the next season at no extra cost. And we can get up to 25% more yields using the same land, water, and fertilizer. So what's next up is we're doing the same thing with corn. Hybrids are very big in corn, as I mentioned before, and it's the #1 crop in USA, and farmers spend 10% of their revenue on hybrid seeds. And this is these advances have attracted a lot of attention. And last month's issue of the journal Nature had a big article describing this that I encourage you to look up now that they've had the background information. And with that, I'll thank my collaborators. Funding from the National Science Foundation, federal funding for basic research is what made all of this possible. And I thank you for your attention. And now I introduce my colleague, Professor Ross from Evolution and Ecology Department.
Great, thanks, Sundar. What I'll talk about today is some work from my lab in quantifying genetic diversity in crops and methods to identify what genetic diversity can be useful for adapting crops to a changing world. Let's see, there we go. By way of introduction, my background is in evolutionary biology and ethnobotany, or the study of how plants and people interact. And my interests revolve around how plants adapt to new environments, especially man-made or human-made environments. My lab uses a suite of computational, statistical, laboratory, and field approaches to understand evolution predominantly of maize or corn and its wild relatives. As we heard in the two previous talks, food crops face a suite of challenges moving forward this century. Most notably, human populations are increasing, so that by end of the century, there will be more mouths to feed, and the climate is changing such that we'll have to produce more food in a harsher or more stressful environment. My lab works mostly on corn. We've heard about corn in the previous talk. One of the reasons we work on corn is it's one of the top calorie products or producing crops in the world. As a sort of context, you think about corn that you buy in the grocery store, that's much less than actually 1% of the market. Most of the corn is used for feeding animals or other purposes, but it's found in lots and lots of other products and foods that we consume. As we saw in Sundar's talk, over the last 60 years or so, there have been dramatic increases in global yields of corn thanks to the efforts of breeders, to the extent that now, for example, there's more acreage of corn grown in China than there is of rice. Unfortunately, in spite of these increases in yield, that's likely not to be good enough. These are results from the UN Committee on Climate Change, where they assessed the vulnerability of different crops to changing climates. And you can see on the map, these are results for maize, that although results vary across the world, across most of the world, you see, and especially in the US, you see a decrease in productivity of corn as climates change, such that by the end of the century, exactly when population sizes will be booming and we'll have more people that we need to feed, their projection is that corn yields will actually, and production of corn will actually decrease by 20 to 30% if we don't do something. In order to do something about it, adaptation and breeding requires genetic diversity. So a breeder can't do much with this field on the top where every plant is exactly clonal replicate of other plants. There's no variation to select on. A breeder or somebody interested in adaptation can work, however, with the field on the bottom, where we have a bunch of different varieties, some of which do well and some of which don't do well in a particular environment. We can select for those that do well in a particular environment of interest, and we can even mix and match genes and create new varieties that are good for environments that we care about. And one of the things my lab is interested in asking is how much diversity do we have? How much genetic diversity do we have? And what part of that genetic diversity do we actually need to adapt crops? One of the ways we do this is with computational approaches to study genetic diversity. Corn, like all other organisms, has DNA. We can sequence these with modern sequencing technologies and identify differences among individual plants. And then we can use those similarities and differences between or among plants to build a sort of family tree or genealogy showing the relatedness of these plants, much like a family tree of people. We, of course, don't do this for 3 plants and a single gene. We're interested in doing this in hundreds or thousands of plants. And we do this not just for one gene, but we do this for all of the genes across the genome. So in the result, we end up with millions and millions of these genealogies for thousands of corn plants. And we can use these genealogies to actually quantify how diversity has changed over time, quantify diversity in a particular population, or see how it's changed as time has progressed. So for example, we can use these results to sort of retrace what diversity and population size have looked like in corn over the last 100,000 years. So corn was domesticated from this wild grass teosinte. The sort of height of this bar here represents the population size or a proxy for genetic diversity. But early farmers only selected a small proportion of these wild grasses to bring into cultivation, resulting in about a 90% decline in diversity as a result of domestication. As maize farming spread across the Americas over the next several thousand years, the population size increased about 50-fold. But then with European colonization about 500 years ago and the concomitant genocide of indigenous farmers, when indigenous farmers perish, unfortunately, their crops perish as well. And we see about a 90% decline in maize populations associated with European conquest and colonization. So a huge loss of diversity. As maize has become a global crop, it's once again expanded and now covers most of the globe. So we see a worldwide spread and a maybe 30-fold increase in population size, although this very recent population size has by no means recovered all of the diversity lost by this set of bottlenecks. Exacerbating these bottlenecks that have lost diversity in corn, modern breeding uses only a tiny fraction of that diversity. So if we look at a breeding program that was important in the modern US, for example, we can actually quantify how many ancestral lineages contribute to that breeding program. And we see even in the 1950s, with the advent of modern breeding, we see that there are only 40 to 50 different lines being used in this breeding program. And as we progress to modern lines produced after the 1980s, this number decreases substantially, such that modern inbred lines or modern hybrids are produced that can trace their ancestry back to as few as 10 different lineages. So a tiny fraction of that diversity is actually being used in modern US breeding. We can also use computational statistical approaches, not just to quantify how much diversity there is, but to learn which of that diversity is interesting. So for example, if we have an environmental characteristic that we care about, like temperature, and we can identify or collect corn plants from different regions of the map that grow in different temperatures, and we can sequence those, we can use a form of artificial intelligence called machine learning and build a machine learning model that then connects the DNA sequence to the environment and learns the relationship between those two. And with one of these models built, you can then do something like pick a new environment, either a current environment, a new place on the map, or a projected environment in the future, put a point on that map, give that to the model, and the model will then predict the DNA sequence that it thinks, the genotype or DNA sequence of corn that it thinks would do best in that particular environment. And so we can do this for lots of different points on the map or points in the future, in future environments. So for example, we can use this to predict what diversity we'll need for particular environments or times in the future. So what I'm showing here is just a plot, a two-dimensional plot of corn genetic diversity. Each blue dot is an actual corn plant that we've sequenced. So this represents several thousand plants. And then the peach dots are what our model has predicted which sequences or genetic diversity we think we need for today's climate. And you can see that those two distributions overlap substantially, suggesting that we've got basically all of the diversity that we need for adapting corn to today's climates. But instead of using today's climates in our model, we can put into our model, for example, the year 2081. And immediately we see there's this huge area on the graph where we have all of these peach circles. These are genotypes that our model predicts that we're going to need this diversity in 2081, but there are no blue dots in that area. There is no current maize genetic diversity in that area of in that part of the graph. We don't have that diversity today. You might ask, well, maybe we'll just, if we leave well enough alone, maybe we'll just get there and sort of current approaches are fine. And we can actually quantify if we don't do any adaptation and we don't do any breeding, how long would it take us to get that diversity that we need for the year 2081? And the answer is about 1,000 years. And since we don't have 1,000 years to wait before the year 2081, this suggests that we're going to need some form of management, active adaptation, or active breeding to identify new genotypes or mix and match the genotypes that we have to create the genetic diversity needed for a particular point in time or particular environment. While modern breeding has taken advantage of or uses a small fraction of the diversity in maize, there's actually a ton of useful adaptive diversity available in maize, thanks to the efforts of traditional farmers, like the one shown on the left, mostly in the Americas, but also in other parts of the world, who have maintained open pollinated populations where every plant is genetically distinct, maintaining lots of diversity. But perhaps more important than just maintaining diversity, they've actively adapted corn plants to a huge array of environments. So shown on the right is a traditional maize field grown at 9,000 feet above elevation in the Andes, grown in traditional agricultural approaches. And here is another traditional maize field grown in the middle of the Mojave Desert with no irrigation. So clearly traditional farmers have done an amazing job of adapting maize or corn to a huge array of environments. And we could potentially use this diversity to adapt modern varieties for future climate change. We're fortunate that over the last many decades, a lot of workers have done some of the very important work for us and collected a lot of these varieties and cataloged them into global seed banks. You may have heard or be familiar with the Svalbard Global Seed Bank shown on the right in Norway, where they collect and store millions of seeds from thousands of varieties of crops to keep them safe for future need for global agriculture. The main seed bank of importance for corn is found in Mexico, run by CIMMYT, and on the left is just a single passageway of that seed bank of their many tens of thousands of different varieties, all of which with these jars of many seeds of each variety. And they've carefully cataloged and they're storing these for safekeeping. And we can sequence these and learn about their genetic diversity and identify genetic diversity that could be useful for adaptation. And so this is one of the avenues that my lab is continuing to work on. In this particular research, in collaboration with the folks at the CIMMYT Seed Bank, we've sequenced several thousand of their varieties that held in their seed bank. And importantly, CIMMYT is now growing many of these varieties in field trials shown in the green triangles on this map. And this allows us not just to have computational sort of abstract statistical projections of what genotypes or what sequences will work well in a particular environment, but we can actually now ground truth those approaches and say, and compare our predictions to what we see in actual field trials, improve our predictions, revamp those, et cetera. So this is a pretty important way of actually testing and improving our computational and statistical approaches. As an example of what we hope we can do with this sort of thing, shown here is a graph of one of our predictions of association of different genes with temperature. So each dot on this graph is a different gene along the corn genome, and the y-axis is how strongly we predict it's associated with temperature. And we can identify regions like this one here with the dot that has the highest association with temperature, and we can look in the genome and say, what's there? And it turns out that in this one is a protein that's a heat stress transcription factor. We were excited by that. It hasn't previously been associated with heat tolerance or stress in corn, but it actually shows response to stress and heat stress tolerance in other crops like tomato. So we can use these sort of approaches to identify potential genes that could help adapt corn to future environments. And these sort of approaches can be used for other crops as well. So once we've identified that diversity, you may ask, how do we get that diversity? We're identifying this in traditional varieties, which Sundar pointed out, don't have very high yield. So we want to move those into modern varieties. And of course, you could do that by crossing, but that takes time and is labor intensive. And more efficient and probably more effective way of moving these is using something like CRISPR or gene editing that Sundar also mentioned. So while we haven't done this for this heat stress transcription factor, we have done this for other genes, like one that we found using similar approaches for flowering time that should allow maize to adapt to different latitudes. If we want to move maize to northern latitudes as climates change, it needs to adapt to different day lengths, and we can do that with genes that affect flowering time. And by CRISPRing this into modern varieties, our colleagues have been able to show that we can indeed get maize that has different sensitivity to day length and different flowering time by moving these genes into modern varieties. So I'll end there, hopefully showing you that using a suite of computational and statistical approaches, we can not only quantify diversity in different breeding populations or diversity through time, but we can use these approaches to identify what genetic diversity is potentially useful for climate adaptation, and hope to, in the future, be able to identify individual genes like this heat stress transcription factor and move those into modern varieties to adapt them to changing climates. Lots of folks to thank, especially folks in my lab over the last 10 years or so, shown on the animated chalkboard, and funding from a number of agencies. And just to emphasize again that none of this would have been possible without funding from federal science agencies, most notably from the National Science Foundation. I'll end there. Thank you.
Well, thank you, Jeff, and thanks to all of the speakers for a remarkable set of talks that were really beautifully integrated. We'll get Sundar back in a moment to answer a number of the questions that are great questions that are already in the chat, as well as the ones that were sent in previously. So I'll try to, I think, just for a quick question to Siobhan first, and then we'll go to the whole panel. There's a question early on here about whether the parasite you discussed is a challenge for the home gardener.
Oh, that is a great question. There is no reason why it won't be a challenge for the gardener. The seeds are going to be prevalent in agricultural soils, but I think we all know that our home gardens are pretty close to those agricultural soils. But if a field is found with the parasite, it has to really lay fallow for a number of years to not be farmed in hopes that it won't spread.
Oh, well, thank you. And thank you for the discussion or reminder about the tomato production in the region. Living up in Woodland, the tomato plant processing plant on Main Street is going full bore, and we see the trucks all the time pulling in.
My favorite time of year.
Yeah. So So a number of the questions are around hybrids and their use in the future, and they also feed into the first question, which is what is the biggest challenge for the future of food and as we enter into the second quarter of the 21st century? So the answer seems to be hybrids, but I don't know if there's other challenges. And then there's a whole thread of questions, both the ones from the pre that were submitted earlier, plus the ones online today about getting traits, valuable traits into this agricultural species. And Jeff just addressed that, as did Sundar, but I think if there's more you want to say or discuss about that. So I guess first, biggest challenges upcoming that might even be beyond the reach of hybrids, or we have to think about different approaches.
Yeah, so I'll try and answer that briefly. So hybrids are basically a vehicle to a method of increasing yields. That doesn't make them automatically immune to striga or to climate change. So these are, but they fit in very nicely. So your hybrids now will be developed using genes identified by Siobhan and Jeff in the future. And again, hybrids are not, although I talk about how we can save seeds for generations, that makes them immediately accessible to the farmer because he's not locked into buying seeds every year. But I've often asked, what about seed companies? Because hybrid seeds are very big business. And the answer is there's yet hope for them because you should be developing new hybrids with the types of genes that Jeff and Siobhan have been working on to meet the future challenges. So I think they all, although they appear to be different topics in the 3 talks, I think they fit together nicely in this sense.
Yeah, I think in addition to, I mentioned climate adaptation, and that's certainly going to be a challenge, even for the best hybrids we have today are predicted not to do particularly well 50, 100 years from now. So there's certainly lots of adaptation and breeding that we need to do, both for disease resistance, pest resistance, and climate change. I think it's also worth highlighting that there's a lot that we can do in terms of agronomics and socioeconomics. So there's a lot of food that's wasted because of shipping policies or because it's rotted or because of economic policies such that it doesn't get to where it needs to go. There's a lot that we can do individually too. 40% or so of corn yield goes to actually feeding animals and not directly to feeding people. So in terms of producing more calories that go directly to people, eating less meat is a good way in that direction too.
Thanks. Yeah, I guess a variation on the question is, Siobhan, you're using forward genetic or mutational analysis to identify important genes for parasites and drought resistance, and Jeff is getting them from natural variation. What's the pipeline look like? We've talked about it a little bit, but it might help people visualize how this works. The pipeline to take that information and get it into a hybrid plant that maybe with Sundar's technology can be made clonally and widely available.
I guess I can take a crack at that. So our
Thank you.
Our research program looks or uses both of those approaches in parallel, right? So we take, for instance, domesticated varieties, and we use genomics to try to identify candidate genes, and then use forward genetic approaches to determine like their pathways and to see how they do what they do. And then in parallel, we also do a lot of work with germplasm from wild species at UC Davis. We have an incredible collection of that. And then we try to mine those genetic resources for sources of adaptive variation. And it's really remarkable what we see in the wild germplasm. For me, in the roots, there's a lot there. Oh, sorry. And then one more thing, when we use those wild species, and so we can breed or breed in those sorts of variation, or we can also use CRISPR to edit genes and to introduce them in that, or to generate these useful alleles in our varieties of interest. And then those in turn can be used in hybrids.
Great, thank you. Taking off from hybrids again, there's a few questions about whether there are particular downsides to hybrids. One here about, nutritionally equal or superior to existing varieties, or other challenges with hybrids. I mean, Sundar's technology gets over a major economic challenge with hybrids, just the production of the seed and its expense. But the crop itself from a hybrid plant, are there any advantages, disadvantages there?
No, this technology is basically once you made the hybrid, how do you keep it going without going back and crossing parents over and over again and charging the farmer every year? But if it's whatever properties were there in the original hybrid, which is already cultivated and accepted, would be preserved in this through this method, because genetically they're identical. So I think there shouldn't be a confusion there that we're not changing anything as such, except the mode of reproduction. So it'll be the same as, and we've done tests to show this actually. We've done grain quality, starch, all these measurements on the rice to show that they're indistinguishable from the
thank you.
From the sexual hybrid.
I might even go further, that at least in corn, I'd say from nutrition, disease resistance, there's sort of no biological perspective in which the hybrids are worse than the inbreds. And the major biological challenge is one that Sundar has overcome. The other challenge is really what we do with them. We went through a really massive economic problem in the 70s because there was one particular hybrid that was grown across most of the US. And that was and that happened to be vulnerable to a new disease that spread around the same time, and we lost something like 40% or 50% of the country's crop because all of the plants were genetically identical. And that's not really a problem with hybrids per se, but how we use them and how we diversify our agriculture.
Thank you, Jeff.
Interesting.
That was actually the result of the method they used to make those hybrids. So they used a particular cytoplasm, which was present in all the lines, because that just was the most convenient way to do it. And it turned out, that's why they stopped. That made it economical, but also susceptible to disease. But yeah, so as I said, it's important to have a variety of hybrids. For rice, it's almost obligatory because rice grows from everywhere, from northern Japan to equatorial regions. You cannot grow the same hybrid in all places. You have to develop customized hybrids that have the same properties for that region. So, so that's one thing. The other thing to also is that although new diseases crop up, they're not like to happen in 2 years. Typically, it takes a while. Somebody in the questions asked about bananas, a famous case of how all bananas were gros Michel, a particular type, for about, and then a disease called Panama will hit them, and now all bananas are Cavendish because bananas are clonal, right? They're not propagated sexually. So, but it should be kept in mind that gros Michel reigned from 1915 to 1965. And Cavendish has been there from 1965 to 2025. So in each case, it was 60 years. I hope that none of these hybrids are going to be around that long. It should be replaced quickly with new genes and with a type that both Siobhan and Jeff are identifying. And that should keep the seed companies happy because there's still a market for what they're making. Sorry.
Right. Well, it's fine. That's great. Thank you. Let's go to a basic science question for a moment. Ron, the question came up about it's early on if you want to look at it in the questions about it appears that more suberin in plant roots is better for drought resistance and disease resistance. Why isn't that just the way plants are already engineered, if you will, or behaving?
Because with every positive comes a negative.
Right. So what's the
So-
negative?
So the negative is that if you have too much suberin, you're basically encasing all of these cells in this like really thick wall, and then they can't grow as well. And so we have to really think about how to find that balance of there being like, it's like Goldilocks, right? Just the right amount of suberin, but also trying to understand, it's also really remarkably responsive, environmentally responsive. So hopefully we can take some cues from nature and see how it's able to quickly change its amounts and respond to the environment and use that as a tool.
All right, thank you. Not knowing much about plants, that's not my area of research. My guess would have been something about the energy cost of just making all that additional polymer and macromolecules and the
It
trade-off.
does require a lot of energy. Yes, correct.
Yeah. So we're picking up in the thread as well, and it was in the earlier questions about public acceptance of genetically engineered plants and whether, and I'll twist it a little bit, and it did come up about whether CRISPR and gene editing is going to be more palatable, or how we talk about it makes it more palatable. There's some thoughts about that.
Okay, I volunteer because I was in India recently in a workshop.
Oh, good.
So
Mhm.
it's made a huge difference. So in India, the government, which is a model for developing countries, and it's a very big one, so in terms of population. So they have actually deregulated gene-edited crops. And 2 varieties of rice have now been approved for release to the farmers. So what happened is, with the GM, genetically modified, bad publicity around it, the regulations were so strict that people stopped developing new crops altogether. Because what is the point if they never get released to go through all this? And so in India, that had a big impact on biotechnology. People just left the field. People who were doing plant transformations moved to other fields of research. And now there's been a revival. It's just amazing to go there and see how much enthusiasm there is. And I think this is really a game changer for crop genetics in the future. That you can do these things, as Jeff showed with the corn, that was gene edited rather than modified, because that's now considered to be acceptable, because there's no foreign DNA going in there, just endogenous DNA that you're modifying, which scientists have been doing anyway. I mean, breeders have been doing, and breeders, by including Native Americans and so on, have been doing a sort of genetic modification anyway for using natural variants.
So was there, with the example in India, public awareness education about the technology, or was it mostly driven by the economics that just need to make more food at a more acceptable price, and the technology became accepted because of the economic drive?
Right now, the farmers are lining up to get these things. So they are the ones who are driving this, not the consumers so much as the farmers who see the benefits of getting more crop production and more. The case of one of the varieties is more drought tolerant, for example, that they've gene edited crop that they're releasing in India. So that's. But of course, the government had a role in the sense that they also deregulated and explained why they were doing this, and they got public comments, and not much has been negative so far. So,
Okay.
And I guess I would add, and I'm hoping that Jeff would also be able to comment on this, because it relates a lot to what you said, which is that we know that gene editing allows us to take advantage of adaptive variation that we've seen, for instance, in wild species, and introduce it in a very targeted way, just recreating what was found in nature, or what could be found in nature, but also taking into account what we have learned over the last decades in terms of how to create a sustainable ecosystem, and the importance of diversity in that context, and the ecology. Jeff, anything to add?
I think there's more still to be done in terms of public education, in terms of gene editing and GMO. I mean, I like to point out that, if we're using gene editing to tweak a particular nucleotide in the corn genome, there's so many corn plants on the planet that actually every nucleotide in the genome mutates something like 500,000 times a year. So there's lots of natural mutation happening all the time. We're just doing this in sort of a controlled way for things that we understand what they're doing.
That's right. So it has been permissible to release in varieties in which there was, say, X-ray radiation or gamma ray radiation to make mutants. You have no idea what's going on in the rest of the genome. Here you have a targeted method where you know exactly what change you're making. I mean, there shouldn't be any, if you're allowing these random rearrangements that are produced by X-rays to be okay, then why should you suddenly balk at a very precise change where you know exactly what's going on? It's just,
More education about the precision and the quality of the tools that we have available to us at this point. We've only got a couple minutes, but the questions that were sent in earlier suggested that we're going to have some students joining us, which is just great. So first one's for Siobhan. You're still systems and synthetic biology advisor. So there's a question about connecting this work to systems and synthetic biology, which I don't think is a very tough question.
No, that's like my bread and butter, right? So if you think about the different scales at which I even described in my presentation, so looking at the genes within the cells, looking at the arrangements of the cells within the roots, looking at the whole root system, we have the opportunity to really understand at all of these different scales, how they interact together, and how they interact together in some likely surprising and unexpected ways. It's important to consider all of these in the context of agriculture and also ecosystems. And so feel free to look into our systems and synthetic biology major, which is just newly offered by the college. And I'm happy to share more with you if you email me.
Thank you for that. I think we're going to wrap up in just a moment. I didn't know if there's any of the questions that you saw or any additional comments the three of you would like to make before we thank you. There is a question about weed control, but
I would just add that branched broomrape and striga are considered to be weeds, noxious weeds by the US. So the idea of using microbes as a way to modulate that is a way to control weeds.
Well, thank you so much. Unfortunately, we're out of time.
Yes, thank you. All right. Yeah.
Yeah.
So I want to thank our speakers, and I'll let Jennifer on in a moment. I really appreciate their time and the presentations that they gave. I want to note that you saw the video from the Institute for Genetics, IGI, down at Berkeley. That was institute's built around Jennifer Doudna and her groundbreaking work in CRISPR technology. But the plant work that they are involved with is with the great faculty in plant science work here on this campus, including Sundar and others. The other organization that has been really important in this field, and it continues to be a driver, you hopefully noticed beyond our federal government, that the Bill and Melinda Gates Foundation has supported all three of these investigators and supports other work on campus as well. So again, thank you to our faculty today. Really wonderful presentations and great integration across the types of work that you do to make for better food in the future. Jennifer.
Thank you so much, Dean Winey. And thank you again to all for joining us today for Under the Microscope, the Future of Food. We hope you found the conversation insightful and inspiring. A huge thank you to our speakers for sharing your exciting work today. Their research is helping to advance the future of sustainable food systems, work that's essential to improving nutrition, supporting environmental resilience, and ensuring food security for future generations. This kind of impact is only possible because of the strong support we receive from our campus community, our partners, and from friends like you. As we celebrate the college's 20th anniversary this year, we invite you to be part of what's next. If you'd like to help us continue driving innovation and discovery, you can scan the QR code on your screen to make a gift or follow us on social media to stay connected. We'll be sending out a short survey, and a recording of today's webinar will go out to all registrants. If you didn't formally register but want to keep hearing from us, just reach out. We'd love to stay in touch. Before we officially wrap up, I'd also like to introduce our development and external relations team, Andy Bowles, Erin Frisia, and Faye Veers. Without them, we wouldn't have such a wonderful webinar today, so thank you to my team. If you have any questions and want to learn more about CBS, we're here for you. So with that, thank you again for your time, your interest, and your support. Until next time.
Yes, thank you.